Okay, good evening everyone. So we'll start today's session. I'll share my screen and you can let me know if it is visible and if I'm audible. Give me a second. Okay. So, yeah. So, good evening everyone. So, today is the fifth live session of this course, RNA Biology. As we know that in these sessions, live sessions, what we do is that we discuss some sample problems related to the course and we try to understand the concept behind this problem so that it's easier for us to solve similar type of other problems. Okay, so that's what we'll be doing in this live session as well. I hope my screen is visible and I'm audible. If I'm not, you can just type it in the chat box or just unmute your mic to say. And we try to keep the sessions as interactive as possible. So if you are having any doubts or if there's anything, then you can just either raise your hand or write in the chat box and we'll get back to you. Okay, so what is the pattern of the class is that I will show you some questions. I'll wait for a few minutes if you are able to solve, and then we'll move on to the conceptual analysis of that question's answer. Okay, so starting with the first question for today. Yeah. Chemical reaction that causes RNA splicing is A. Condensation, B. Hydrogen bonding, C. Coupling, and D. Transacidification. So I would wait for a few minutes where anyone of you want to answer. You can go ahead and then we'll move on to the answer. So if you know the answer, you can write in the chat box and then we'll move on to the discussion. I'll wait for a few more minutes. Okay, so the right answer to this will be transesterification. So before moving on to the answer, we'll discuss the background a little bit. So we have talked about ribozymes in most of the previous classes. Again, a little bit of background. Ribozymes are basically RNA processing catalytic. They possess RNA processing catalytic activity. Okay, and increases the rate and specificity of phosphodiester bond cleavage, peptide bond synthesis. And there's a widespread occurrence in nature from viruses to humans, right? So this is in 1989 Nobel Prize in Chemistry was awarded to Sidney Altman and Thomas Sheck for the discovery that RNA in living cells is not only a molecule of heredity but also can function as a biocatalyst. So then there are naturally occurring ribozymes. So we have a ribozyme RNA tRNA, which is catalytic functional peptide bond formation, and the biological formation will be protein synthesis. Ribonuclease P will have phosphodiester bond hydrolysis function, and the maturation of transfer RNA will be the biological function. Then we have cell splicing where phosphodiester bond hydrolysis occurs, and ligation group 1 and group 2 introns, and the function is removing introns from precursor messenger RNA. Hairpin ribozyme, hammerhead, HTV, or phosphodiester bond hydrolysis and RNA virus replication pathways involved in. So the structural features affect how RNA can function. So RNA contains only 4 unique nucleotide bases compared to 20 amino acids found in proteins, like small repertoire of functional groups in genes, right? In RNA, the high density of negative charges, the localization of bases in the interior of duplexes, like amino acid side chains, are directed outward from the polypeptide backbone. Nevertheless, the mechanisms of catalysis are diverse and exploit. Metal ions, acid-base mechanism, that is using nucleobases, small molecule metabolite as cofactor, substrate like tRNA assistance, and usually ribozyme combines several of these strategies. So the catalytic strategies appear to be similar. RNA as well as protein enzymes use acid-base groups in metal ions to activate nucleophiles and to stabilize developing charge on the leaving group. Ribozyme also requires formation of a specific secondary and tertiary structure of RNA by base pairing of complementary region. Specific primary structure of certain regions is also necessary. Now, some ribozymes can speed up the rate of reaction 10 to the power 3, 10 to the power 11 times. HDV ribozyme leaves the phosphodiester bond as fast as RNAs. If you talk about metalloribozymes, ribonuclease P, RNase P catalyzes site-specific hydrolysis of precursor tRNA, which is essential for the formation of mature tRNA. So catalytic activity depends on the presence of divalent cations like Mg2 plus Mn2 plus. And large ribozyme composed of both RNA and proteins. However, RNA moiety alone is the catalyst. Now, large introns that are able to splice out themselves like greater than 200 nucleotides, and in bacteria as well as eukaryotes like pRNA of protease 1 tetraamine primary transcripts of the mitochondrial genes of yeast and plants. If we talk about splicing and introns are segments of non-coding RNA that are interspersed among the regions of mRNA that code for protein, like exons. Now, prior to translation, introns must be removed to form a mature mRNA. If you see, we have the genomic DNA, then the pre-mRNA, and then splicing spliced mRNA. Now, unlike common introns, cell-splicing introns can splice themselves out of pre-mRNA without the need for the spliceosome, like complex of RNA and proteins, enzymes like helicases. Now, although cell-splicing introns can remove themselves from RNA in the absence of any protein in vitro, in many cases in vivo, cell-splicing proceeds in the presence of certain proteins that increase the efficiency of splicing, stabilize the correct structure of RNA. A cell splicing introns mediate only one round of RNA processing, unlike other protein enzymes. Now, there are a group 1 introns. Cell splicing is initiated by the nucleophilic attack of 3' OH of an exogenous guanosine bound by hydrogen bonds on the phosphodiesterase bond. And then we have group 2 introns. Nucleophile attack is realized by 2' OH of a specific adenosine within the intron. Now, metal ions like Mg2 plus Mn2 plus are proposed to promote the formation of the correct active structure, correctly position the substrate active with the nucleophile by deprotonating the 2' OH of guanosine, and it stabilizes the negative charge as well. So if we talk about the group 1 introns, the 3' OH of an exogenous G attacks the phosphodiesterase bond at the 5' splice site. This bond is being cleft. G fuses to the 5' end of the intron, and first transesterification is what is happening right now. Then the free 3' end of the exon attacks the bond at the 3' splice site. This fuses the two exons and releases the intron, the second transesterification. So if you see the schematic over here for group 1 intron, they have G-nucleotide G nucleotide binding site. G attacks the phosphodiesterase bond at the 3' splice site, cleavage between 3' and 5' end of exon and 5' the end of the intron. The terminal 3OH of exon 1 attacks and leaves the phosphodiesterase bond at the 3' splice site. A new bond is formed between the two exons and intron is released. And when the internal A attacks the phosphodiesterase bond at the 5' splice site, this is the schematic over here. Now, what is the importance of being folded? Specific primary, secondary, and tertiary structure is necessary for recognition of the guanosine binding site and recognition of the attacks of the splicing. Right? This is the 5' splice site, the splicing site recognized by guanosine and attack of the first attack. Site of the first attack and base pairing and all of that. This is RNA hairpin. Now, cell splicing introns mediate only one round of RNA processing, unlike protein enzymes. But once a group 1 intron has been spliced out, it can act as a real enzyme. It can repeatedly recognize a complementary sequence of another RNA molecule by the internal guide sequence, attacking the 3' OH of the bound G-nucleotide and catalyzes its cleavage. So this is how the group 1 intron after being spliced out will look like. This is ribosome attacking the RNA substrate. Now we can change the IGS and thus generate tailor-made ribosomes that cleave or destroy RNA. Cleave or destroy RNA molecules of our choice. Candidate method for human therapy. So currently synthetic ribosome that destroys mRNA encoding the receptor of vascular endothelial growth factor is being readied for clinical trials. VEGF is a major stimulant of angiogenesis, and blocking its action may help starve cancers of the blood supply. Now, small ribosomes of virions and satellites, hairpin, hammerhead, and HTV hepatitis delta virus. Now, small RNA viruses or RNA molecules, their multiplication depends on the mechanisms of the host cell and on the co-infection of a host cell with a helper virus. So, ribosome is a part of the larger RNA, virion or satellite. This being replicated by the host RNA polymerases. So, the product of the replication is being self-cleft by ribosome activity into unit length RNA molecules. So, nucleophilic attack at the 2' OH of the neighboring 3' phosphate forming 2' 3' cyclide phosphate. Then probably an acid-based mechanism, 2' OH is activated for a nucleophilic attack by abstraction of a proton by a basic group B. Another proton is donated by an addition acid S to stabilize the developing negative charge on the leaving group oxygen. Now, in HDV, cytosine acts as an acid to protonate the living group and divalent cation activates the nucleophile. Now, hammerhead and hairpin ribosomes can be found in several satellite RNAs associated with RNA plant viruses. RNA is a human pathogen. Human infection of HDV with HBV is more severe than HDV alone. Now, what are the elements of bacterial RNA that control gene expression where bending of small molecules, ribozymes, or amino acids or nucleotides? Nucleobases. What is GLMS ribozyme? Located in the 5' untranslated region of mRNA encoding glucosamine 6 phosphate synthetase in the presence of GLCN6P, which lays its own mRNA, which downregulates the production of synthesis. Now, mechanisms of ribozymes which catalyze translation. So, all informational metabolite binding includes a conformational change in RNA that affects transcriptional termination and translation mechanism. So, mechanical reading at me. Chemical GLMS, GLC, and 6P amine might serve as an acid to activate the living group cleavage, right? So if you see, this is the role of peptidyl transferase ribosome, the translation. So peptidyl transferase activity can be enhanced by protein L27. However, seen in the absence of this protein, reduced activity can still be observed. So although the protein facilitates peptide bond formation, it is not necessarily essential for peptide transferase activity. Now, how does RNA catalyze peptide bond formation? So base pairing between the CC end of the triRNAs in the P and A sites and 223 sRNA help position the amino acid group of amino acid tRNA to activate the carbonyl group of the going polypeptide. So proton transfer from the amino acid of amino acid tRNA at 2' adenosine phosphate from the terminal of C or the CA of RNA to its 3' side. RNA initially served both as genetic material and the catalyst. Later, catalytic actions of many RNA molecules were taken over by proteins. Now, cationic clays such as montmorillonite can promote the polymerization of RNA-like polymers into RNA chains. So, RNA is the primary structure of life. DNA and proteins are later refinements. Now, cofactors used by ribosome include vitamin B12, FNN, glucosamine 6 phosphate. Some of them are used by protein enzymes for oxidation reduction, CC1 formation. So we also know that RNA molecules capable of something like this. Also, some of them persisted up to now. So what do we have? Protein catalysts. So group 1 intron active site is mechanistically equivalent to DNA and RNA polymerase. So what selective pressure led to the current protein-based system for replication and transcription? The reason might be much greater, like fidelity, processivity, reaction rates, functional repertoire, all of these other reasons. So I talked about the initiation special promoter sequences from -10 and -35. Sigma factor binds to promoter, helices fit into the major groove. A special promoter sequences, as I already told in initiation, sigma factor binds to anyone, it is in major group, and you get several transcription factors assemble and bind to promoter. Now, regulation of transcription. Cis and trans regulates our factor sequences, upstream data box enhances, etc. And cytose proteins that bind to promoter made by other faraway allele genes. Now, chromatin structure also regulates transcription. Histones prevent transcriptional factors from binding. In interphase DNA is 30 nanometer fiber phase wound thoroughly around nucleosomes. The structure of RNA to RNA transcript and adds a long time sort of AIs or poly A tail. So critical sequence are in the intros 5' GU branch point A, 3' AG or AKGU. So details of intron removal from PMRF cleavage at 5' exogenous function and the version of every pi girl's father. Cleavage of at 5' exon intron junction and formation of lydate structure. Then we see the branch point sequence. So you see the lydate structure is like this. This is the branch point sequence. And cleavage at the cleavage at the 3' exon intron junction and ligation of the two exons. Okay. And branch junction structure involving unusual 2' 5' phosphodiester bond between the G nucleotide from 5' junction and the branch point sequence. Now, both the 5' splice site and the branch site are checked before any splicing reactions occur. The sequence must pass both identity checks. So U1 is replaced by U6 at the 5' SS and BBP SF1 is replaced by U2 at the branch site. Splicing mechanism U2 and U6 hold the pre-mRNA in a way that juxtaposes the 5' S and the travel branch point. Now, what are splicing enhancers? They may also be special splicing enhancers sequencing the exons. Proteins and RNA bind to these sites and define the exons. So, this is adapted from Pre-Polar RNA Unshaw Cell Biology book. SRNPs, SR proteins and splicing factors associated with phosphorylated CTD, C-terminal, which has a cross-exon evolution complex. Splicing errors can lead to disease, spinal muscular atrophy. Okay, SMN1, SMN2 is a huge example. I'll also explain this in one of the other questions. Mutation is in SMN2 causes a skipped exon in the mRNA. A group 1 introns in RNA first to utilize the specific splicing reactions. For example, of an RNA enzyme, what does it mean? Important for ideas about the origin of life, often called the RNA world, etc. Polymerase 1 transcripts ribosomal RNA. Ribosome is used to make proteins as two subunits, large and small. Large subunit is composed of RNA, 28S, 5.8S, and 5S, and many proteins. Small subunit has 18S rRNA and many proteins. So in eukaryotes, all of these are adjacent with sequence repeating hundreds of times. So RNA genes and RNA production in E. coli. In case of poultry, makes tRNA and 5S rRNA transfer RNAs are to make the proteins because they bring the correct amino acid. Each tRNA has a unique sequence and slightly different shape. All are plus minus clover leaf. If we talk about transfer RNA, anticodon at one end recognizes RNA sequence, amino acid attachment site at the other. Always CCL added after transcription. often have several modified bases, uracil, ribo, ribothamidine, etc. So these modifications have happened post transcription. Is the closest clover leaf structure for yeast RNA and tRNA. A promoter of 5S and tRNA are within the gene sequence upstream. So here's a mature mRNA sequence and this is the DNA sequence. Does the DNA maintain the intron? Where does it bind the splice sites and not the branch point? Does it fit the pot synthesis sequence? Model for intron lysosome. Some genes have alternative splicing such that different exons are used in different conversations or issues. Troponin. So mature mRNA consists exons 1, 2, and 4 in smooth muscle. Mature mRNA contains exons 1, 3, and 5 in the other tissues. RNA editing. In some case species, the final RNA molecule is much longer than the corresponding DNA sequence. Other bases are added after the transcription, like fat resolution, like how? Example from trypanosome. Okay, this is a comparison of the DNA sequences of the cytochrome oxidase subunit gene in the protozoans. One example is spinal muscular atrophy, one of the most common genetic disorders. In humans, such an essential gene called SM1 has been duplicated and compared to that humans, but as well as the mouse. SM2 encodes a protein identical with SM1 expressed to much lower level because silent mutation is one and exon interfering with the kind of the protein resulting in exon skipping and in most but not all NS72 RNAs. Spinal atrophy occurs when the mutations occur in SMN that fact inactivates efficient SMN protein is expressed from SM2 to allow embryonic and fetal development. However, during childhood, spinal cord neurons die because of insufficient SMN, resulting in the disease. This is pre-mRNA processing. How you tell us for the mRNA splicing. RNA splicing puts the pieces back together. Most mammalian genes contain more than one intron. Nucleosomes are preferentially bound to exons, where introns are mostly devoid. Exons are therefore marked at the DNA level by nucleosome positioning, which may act as speed bumps for DNA polymerase, RNA polymerase 2. There are currently two opposite theories of intron again. Intron's early theory proposes that introns already exist in the progeny and to facilitate the expression of the first genes. The intron's late theory, on the other hand, he asks that genes of the propaganda were late and after the emergence of eukaryotes. This will not no decisive solution to the debate, and each of these theories has supported kernels that have not been sacrificed. So, what are the advantages of intron alternative splicing, new exons, new introns, shuffling? Muons will have protein demands. Many genes have evolved and similar exons are shared. Some shuffling before and after. The length varies from 1,04,000 highlights in the human genome, consider about 10% of the human genome. or mostly and propagate the IST is in the genome through retroposition or RNA intermediates. Splicing requires RNA and protein in regulators. It has a 68 dynamic structure, a large subunit consisting of 150 proteins, 5 small subunits. RNA is assembled with proteins to form um, flour. Assembly of spliceosome requires ATP. The spliceosome contains siRNAs, siRNAs, and many other proteins, totally about 300 subunits. This makes it the most complicated macromolar machine known to date. But why is spliceosome so extremely complicated? If it only catalyzes such a straightforward reaction as in intron deletion. Even more, it seems that some introns are capable to exercise themselves without aid of any protein. So I have all those 300 minutes. No one knows for sure, but there might be at least 4 reasons. Defective mRNA causes a lot of proteins to the cell, so you might subunits might assure splicing and erection. Splicing is coupled to nuclear transport. This requires additional proteins in splicing. Heat is coupled to transcription, and this might require more additional access of proteins. Many genes are replaced in several ways, and it also might mean additional factors. So you have 5 small RNAs U1, U2, U3, U4, and U5 bind to proteins to form SNRP, RNP, which the mammalian SNRP consists of the 10 different proteins such as isostereonucleotide RNA molecules. Even all of the proteins, the same proteins are common to all SNVRNAPS, whereas the other genus is with least contains conserved sequence complementary to splice of a nuclear mRNA intron. Secondary structure of mRNA is transcription with 5' site interaction with branch site, interaction between U2 and U6, SM binding site, which was so base pairing between pre-RNA, siRNA, and U1 siRNA and U2 siRNA early in the cycle. I explained demonstrates that the base pairing between U1 and the 5' acid is in pre-RNA's context. So there is a model of spliceosome mediated splicing of the pre-RNA. So is illegal. Go to the slides. Okay. Already discussed all of this. Okay. So the correct answer will be transition. If anybody has any doubts in this question, we can take the question now or we can do it later after I have finished all the questions. So anybody has any doubts? I'll give you some time to figure out if you have any doubts. every doubt in this question and move on to the next question for today. So we'll move on to the next question for today. Which of the following results from mutations in splicing factors? A. Duchenne muscular dystrophy. B. Cystic fibrosis. C. Spinal muscular atrophy. D. Dementia and Parkinsonism. I'll wait for a few minutes. You can decide and try to answer. Then I'll move on. You can try answering onto the concepts. Okay, starting off with the question, the answer will be spinal muscular atrophy. Anybody knows why? Otherwise, I'll move ahead. Okay. So spinal muscular atrophy is basically a broad phenotype spectrum ranging in the age of onset, like birth to adulthood, severity clinical course. So types of SMA are primarily distinguished by the severity of muscle weakness and age of system onset. Type 1 and 2 are more severe. So what basically happens in spinal muscular atrophy? It's a rare genetic condition that affects the motor neurons in the spinal cord, causing progressive muscle wasting and weakness. It can impact people of all races and genders, and is usually diagnosed in infancy or early childhood. SMA is caused by a gene change in both copies of the SMN1 gene and is inherited in an autosomal recessive pattern. Okay. So basically it affects the entire central nervous system, peripheral nervous system and voluntary muscle movement. So most of the nerve cells that control muscles are located in the spinal cord. So which accounts for the word spinal in the name of the disease. SMA is muscular because its primary effect is on muscles which don't receive signals from these nerve cells. Atrophy is the medical term for getting smaller, which is what generally happens to muscles when they are not stimulated by nerve cells. Now, SMA involves the loss of nerve cells called motor neurons in the spinal cord and is classified as a motor neuron disease. It is the most common form of SMA where there is a wide variability in the age of onset, symptoms, and rate of progression. In order to account for these differences, chromosome 5 related SMA, which often is autosomal recessive, is classified into types 1 through 4. So the age at which SMA symptoms begin roughly correlates with the degree to which motor function is affected. So the earlier the age of onset, the greater the impact on motor function. Children who display symptoms at birth or in infancy typically have the lowest level of functioning or type 1. Later onset SMA with a less severe course type 2 and 3 and with teens or adults type 4 generally correlate with increasingly higher levels of motor function. Now what causes SMA or spinal muscular atrophy? So chromosome 5 SMA is caused by a deficiency of a motor neuron protein called SMN for survival of motor neuron. So this protein, as its name implies, seems to be necessary for normal motor neuron function. SMN plays a pivotal role in gene expression in motor neurons. Its deficiency is caused by genetic flaws or mutations of chromosome 5 in a gene called SMN1. And the most common mutation in SMN1 gene within patients diagnosed with SMA is the deletion of a whole segment called exon 7. Now, neighboring SMN2 genes can in part compensate for non-functional SMN1 genes as there is 99% identity between these two genes. So other rare forms of SMA are caused by mutations in genes other than SMN1. So what are the symptoms of SMA? SMA symptoms cover a broad spectrum ranging from mild to severe, and the primary symptom of chromosome 5 related SMA is weakness of the voluntary muscles. So the muscles most affected are those closest to the center of the body, such as those of shoulders, hips, thighs, and upper back. The lower limbs seem to be affected more than the upper limbs, and deep tendon reflexes are decreased. The special complications occur if the muscles used for breathing and swallowing are affected, resulting in abnormalities in these functions. So, if the muscles of the back weaken, spinal curvatures can develop. So, there is a great deal of variation in the age of onset and developed motor function achieved in chromosome 5 related SMA. These are roughly correlated with how much functional SMN protein is present in the motor neurons, which in turn correlates with how many copies of SMN2 genes a person has. sensory, mental, and emotional functioning are entirely normal in chromosome 5 SMA. Now, some forms of SMA are not linked to chromosome 5 or SMA deficiency. These forms vary greatly in severity and in muscles most affected. So, while most forms like chromosome 5 related form affect mostly the proximal muscles, other forms exist that affect mostly the distal muscles, like those farther away from the body center, at least in the beginning. So, what is the progression of SMA? In chromosome 5 related SMA, the later the symptoms begin, and the more SMN protein there is, the milder the course of the disease likely to be. So, while in the past infants with SMA typically did not survive more than 2 years, today most doctors now consider SMN related SMA to be a continuum and prefer not to make rigid predictions about life expectancy or weakness based strictly on age of onset. So SMA is the most common genetic cause of mortality in infants. What is the status of research on SMA? Research has focused on strategies to increase the body's production of the SMN proteins lacking in the chromosome 5 related forms of the disease. Approaches include methods to help motor neurons survive in adverse circumstances. Okay. So this is all about spinal muscular atrophy. So basically autosomal recessive disease affecting the lower motor neurons in the spinal cord and brainstem, resulting in progressive motor weakness and atrophy. This is type 1, type 2 and all of that. So this image is from this particular page. You can see the cement to centromeric copy is in one telomeric copy and approximately 15% exon 7 inclusion and all of that, which will lead to a truncated S7 protein non-functional, or the telomeric copy will lead to approximately 15% 100% functional S7 protein. Okay, so this is a process of diagnostic algorithm for the spinal muscular atrophy. Sorry, this will not go into too much details of the factual data. Okay. So there are five subtypes of SMA. Healthcare providers classify them based on the age of onset, as I already told you, as well as the severity and life expectancy. Subtypes include SMA type 0 or the congenital SMA. It's a rare subtype that affects the fetus before birth. Pre-birth, there are typically decreased fetal movements. At birth, infants with type 0 have severe muscle weakness and typically go into respiratory failure, and death usually happens at birth or within the first month of life. Then we have SMA type 2. About 60% of SMA cases are type 1, type 1. Severe SMA, 60% of the cases also called Werdnig-Hoffmann disease. Symptoms arise within the first 6 months of life and include limited head control and decreased muscle tone or hypotonia. Infants with type 1 SMA also have difficulty swallowing and breathing. So, without breathing support, children with type 1 SMA die before the second birthday. SMA type 2 or intermediate SMA, symptoms of type 2 SMA, also called Dubovic's disease, appear between 6 months and 18 months of life. Symptoms include hypotonia, worsening muscle weakness, which tends to affect their legs more than their arms. Children with type 2 SMA may be able to sit up but can't walk. So around 70% of the people with type 2 will survive until 25, with some surviving into their 30s. Respiratory issues are the major cause of disease and death. Then we have SMA type 3 or mild. Symptoms of type 3 SMA, also called Gilbert-Wallender disease, appear after a child's first 18 months of life. Type 3 symptoms include lower limb muscle weakness, leading to difficulty walking. People with type 3 SMA don't tend to develop breathing issues, and it typically doesn't affect life expectancy. We have SMA type 4 or adult. This is the mildest form of SMA. It doesn't appear typically until after the age of 21. Muscle weakness symptoms progress slowly, so most people with type 4 remain mobile. It typically doesn't affect life expectancy. So all the SMA is thought of as uncommon. It's the second most common severe hereditary disease of infancy and childhood after cystic fibrosis. Researchers estimate that it affects between 1 in 6,000 to 1 in 11,000 type births. It's about twice as common in white and Asian people as it is in black and Hispanic. So, what are the symptoms? In general, the main symptom of SMA will be muscle weakness, typically the muscles closest to the center of your body. But the symptoms may vary based on the type as well. Decreased fetal movements, for SMA type 0, arthriodermopsis, decreased muscle tone or hypotonia, severe muscle weakness, severe breathing issues. What type? SMA type 1 symptoms are limited head control, hypotonia, lack of reflexes, inability to sit without support, abnormal breathing pattern, and a bell-shaped chest, difficulty swallowing, which can lead to growth issues, facial muscle weakness. This develops later on in the condition. Symptoms of SMA type 2 include hypotonia, ataxia, progressive muscle weakness that affects your child's legs more than their arms, scoliosis, muscle weakness in your child's chest, which can result in restrictive lung disease, jerky movements in your child's hands, polymyalgia, stiffening of your child's jaw, joint due to bone fusion, joint contractures. Symptoms of SMA type 3 mainly involve progressive muscle weakness that affects your child's legs more than their arms. And the main symptom of SMA type 4 is mild leg weakness. What causes spinal muscular atrophy? It's a genetic condition, as I already said, which means you inherit genes from your biological parents that cause the condition. And the mutations in the SMN1 gene causes all types of spinal muscular atrophy. The number of copies that you have of the SMN2 gene alters the severity of the condition. Here, the SMN1 gene produces SMN protein. Motor neurons need this protein to survive and function properly. If you have SMA, your body doesn't make enough SMN protein, so your motor neurons shrink and die. As a result, your brain can't control voluntary movements, especially motion in your head, neck, chest, and legs. And the SMN2 gene also produces a small amount of SMN protein. A person may have up to 8 copies of an SMN2 gene. Having multiple copies of the SMN2 gene typically leads to less severe SMN symptoms because the extra genes make up for the missing SMN1 protein. What is the inheritance pattern of SMA? You inherit is SMA in an autosomal recessive pattern, which means both of your biological parents pass on mutations in the SMN1 gene. In most cases, the biological parents of someone with an autosomal recessive condition each carry one copy of the mutated gene. But these carriers typically don't have symptoms of the condition, and general population mutations of the SMN1 gene are common. Among white people, 1 in 50 may be carrier. In rare cases, you may inherit an SMN1 gene mutation from one parent and acquire a new mutation in the other copy of the gene during embryonic development. Okay. So you see this is the CSF flow, central system fluid, and spinal fluid, and the antisense oligonucleotide is injected into the intrathecal space. Okay, so we all have autosomal recessive pediatric neurodegenerative disorder, deletions in the FQ13 SMN1 gene, that's chromosome, survival of the motor neuron gene, It's important for motor neuron health and survival and progressive loss of alpha motor neurons in the anterior horn cell of the spinal cord. Okay. Incidence is 1 in 10,000. Carrier frequency about 1 is 35 in the Caucasian population. Maybe even higher because we may miss asymptomatic individuals, embryonic lethal subjects. The truncated SMN protein is unstable and non-functional. The SMN2 gene allows for rescue from embryonic lethality, less efficient. Each copy produces about 10 to 15% full-length protein compared to SMN1 gene. So these are the symptoms of SMA type 2. I already discussed all of this. SMA type 3 is also called Gugelberg-Welander disease. These are the symptoms of SMA type 3. Just go through. I have already spoken about all these. So these are DNA testing for SMA. That will be the first line of defense. I think molecular genetic PCR-based testing. 2-3 weeks needed for the result. Now it's much quicker because of medical advances. 95% sensitivity and specificity. Homozygous deletions of SMN1. Less used as first line, possibly more in later onset cases. Okay. Drug development. Therapeutic approaches. Transmission SMN2 activation by gene promoter. Restorative splicing, translational activation and stabilization of the FLSMN protein, suppression of the SMN2 stop codon, elongate protein. Clinical trials, we don't need to get into all these details. So coming back to the answer, these are the references for all the details. So coming back to the answer, that is establish that the following results between the splicing factors will be spinal muscular atrophy. Okay. Anybody has any questions regarding this? We can discuss or we move on to the next question for today. Okay. So we'll move to the next question for today. If nobody has any doubt in this question, mRNA maturation is dependent on DROSH drosha, DGCR8, DICER, DICER, or RISC. I'll give you some time. If you can figure out the answer, then we'll move on to the answer, discussing the answer. Okay, so the right answer for this question will be dicer, DICER. Okay, so if we start from the beginning, okay, this is a very information heavy slide. So if you see, scientists have gotten their first detailed look of the molecular structure of an enzyme that nature has been using for years to help silence unwanted genetic messages. So a team of researchers with Berkeley Lab and University of California, Berkeley used X-ray crystallography and ALS beamlines to determine the crystal structure of dicer. It is basically an enzyme that plays a critical role in process known as RNA interference. So the RNA enzyme dicer enzyme, is able to snip a double-stranded form of RNA into segments that can attach themselves to genes and block their activity. So with this crystal structure, the researchers learned that dicer serves as a molecular ruler with a clamp at one end and a cleaver at the other end, a set distance away, that produces RNA fragments of an ideal size for gene silencing. RNA ribonucleic acid has long been known as a multi-purpose biological workhorse responsible for carrying DNA's genetic messages out from the nucleus of a living cell and using those messages to make specific proteins in a cell cytoplasm. Now in 1998, however, scientists discovered that RNA can also block the synthesis of proteins from some of those genetic messages. This gene silencing process is called RNA interference, and it starts when a double-stranded segment of RNA encounters the enzyme dicer. Dicer cleaves dsRNA, double-stranded RNA, into smaller fragments called short interfering RNAs and microRNAs. Dicer then helps load these fragments into large multi-protein complex called RISC for RNA-induced silencing complex. RISC can seek out and capture messenger RNA molecules, RNA that encodes the message of a gene, with a base sequence complementary to that of its siRNA or miRNA. This serves to either destroy the genetic message carried by the mRNA outright or else block the subsequent synthesis of a protein. Until now, it has not been known how dicer is able to recognize dsRNA and cleave those molecules into products with lengths that are exactly what is needed to silence specific genes. Now, the Berkeley researchers were able to purify and crystallize the dicer enzyme from Giardia intestinalis, a one-celled microscopic parasite that can infect the intestines of humans and animals. This dicer enzyme in Giardia is identical to the code of a dicer enzyme in higher eukaryotes, including humans, that cleaves dsRNA into dense of about 25 bases. So in this work, the researchers describe a front view of the structure as looking like an axe. On the handle end, there is a domain that is known to bind to small RNA products, and on the blade end, there is a domain that is able to cleave RNA. Now, between the clamp and the cleaver is a flat surface region that carries a positive electrical charge. Now, the researchers proposed that this flat region binds to the negatively charged double-stranded RNA like biological Velcro, enabling dicer to measure out and snip specified lengths of siRNA. So when you put the clamp, the flat area and the cleaver together, you get a pretty good idea as to how dicer works. The research team is now using the structural model to design experiments that might reveal what triggers dicer into action. In addition, one size does not fit all for dicer. Different forms of dicer enzyme are known to produce different lengths of siRNA, ranging from 21 to 30 base pairs in length or longer. So, having identified the flat surface positively charged region in dicer as the ruler portion of the enzyme, the researchers speculate that it may be possible to alter the length of a long connected helix within this domain to change the lengths of the resulting siRNA products. Researchers would like to see what happens when you take a natural dicer and change the length of its helix. Okay. So, RNA interference genetic process in which the targeted genes can be turned off. So, the necessary step in many fundamental biological events: genome rearrangement and stem cell differentiation, brain development, viral defense. You can infer a gene's function by observing effects of switching it off. Can also be put to practical use, treating cancer, improving crop pest resistance. Lysin enzyme known to play a key role in RNA interference. The details of how it functions have remained unclear. Okay. Now, what is gene silencing? So, it's basically a technique that aims to reduce or eliminate the production of a protein from its corresponding gene. It generally describes the switching off of a gene by a mechanism other than genetic modification. Okay, that is a gene which could be expressed under normal circumstances is switched off by machinery in the cell. It occurs when RNA is unable to make a protein during translation. Gene silencing is same as gene knockdown, but is totally different from gene knockout. When genes are knocked down, their expression is reduced. Parent controlled genes are knocked out, they are completely erased from organism's genome and thus have no expression. Now, to deepen the pigmentation in petunias, introduction of transgenes homologous to endogenous genes often resulted in plants with both gene suppression or co-suppression, resulted in degradation of the endogenous and transgene mRNA. Okay. Discovery of the mechanism of RNA interference by dsRNA by Professor Andrew Fire. So, how does it work? This is accomplished by binding a specific strand of RNA to an existing mRNA strand. The mRNA creates a copy of the DNA strand. By binding the RNA to the mRNA is prevented from replicating that portion of the DNA. Specific genes can be targeted and prevented from replicating into new DNA strands. Now, what are the types of gene silencing? Genes are regulated at either the transcriptional level or post-transcriptional level. Therefore, silencing can be induced either at transcriptional level or post-transcriptional level. And there are mainly two types of gene silencing: transcriptional gene silencing and post-transcriptional gene silencing. So, in the first case, the promoter is silent, in the second case, promoter is active. The genes are hypermethylated in the promoter region, and second case, genes are hypermethylated in the coding region. And the purpose for both is viral immunity. Now, one second. Is a result of histone modifications creating an environment of heterochromatin around a gene that makes it inaccessible to the transcription machinery. Genomic imprinting is a genetic phenomenon by which certain genes are expressed in a parent of origin specific manner. It is an inheritance process independent of the classical Mendelian inheritance. Genomic imprinting have been demonstrated in insects, mammals, and flowering plants. A paramutation is an interaction between two alleles of a single locus, resulting in a heritable change of one allele that is induced by the other allele. Paramutation was first observed by the effect it had on the color of corn kernels in maize plants. Now, position effect is the effect of expression of a gene when its location in the chromosome is changed, often by translocation. This has well been described in Drosophila with respect to eye color and is known as position effect variation. Now, if we talk about RNA-directed DNA methylation, is an epigenetic process first elucidated in plants whereby small double-stranded RNAs are processed uh, to guide methylation to complementary DNA loci in the model plant organism Arabidopsis thaliana. Transposon silencing is a form of transcriptional gene silencing targeting transposons. Is a product of Western modification that prevents the transcription of the area of DNA. But the jumping of transposon generates genomic instability and causes extremely deleterious mutations. So, transposable element insertion have been linked to many diseases, including hemophilia, SCID, predisposition to cancer. Unfortunate insertion of transgene into a transcriptionally inactive part of the genome. When an insertion of any transgene does not show activity as per desire, and this is because of its instability. Loss of transgene stability is because of gene silencing. Explore fruit softening tomato by reducing expression of polygalactolinase enzyme. Now, what is post-transcriptional gene silencing? The ability of exogenous or sometimes endogenous RNA to suppress the expression of the gene which corresponds to the mRNA sequence. It is a post-transcriptional process triggered by the introduction of double-stranded RNA which leads to the gene silencing in a sequence-specific manner. First evidence comes from studies on nematode Caenorhabditis elegans. Further analysis in fruit fly Drosophila melanogaster, also known as post-transcriptional gene silencing, post-suppression or quelling. Now, during RNA interference, double-stranded RNA is cut into short double-stranded RNAs by an enzyme called dicer. These then base pair to an mRNA through an dsRNA enzyme complex. This will either lead to degradation of the mRNA strand. Highly specific process, very potent activity. So far, only been seen in eukaryotes. Evidence 30% of genome is regulated by RNA1. RNAi is pathways guided by siRNA, small interfering RNA, that have an integral role in the phenomenon of RNA interference, a form of post-transcriptional gene silencing. RNA interference 21-25 nucleotide fragments which bind to the complementary portion of the target mRNA and tag it for degradation. A single base pair difference between the siRNA template and the target mRNA is enough to block the process. Each strand of siRNA has a 5' phosphate termini, 3' hydroxyl termini, 2 nucleotides, 3' overhangs. miRNA originate from capped and polyadenylated full-length precursors. Hairpin precursor 70 nucleotide, mature miRNA 22 nucleotide. miRNA originates with ssRNA that forms a hairpin secondary structure. It regulates post-transcriptional gene expression and is often not 100% complementary to the target. And also miRNA help to regulate gene expression, particularly during induction of intrachromatin formation. It serves to down regulate genes pre-transcriptionally. Okay. Dicer RNA is free like DS RNA specific ribonuclease enzyme involved in the initiation of RNA interference. It is able to digest DS RNA into uniformly sized small RNAs. Dicer family proteins are ATP dependent nucleases. RNase 3 enzyme acts as a dimer. Loss of silencing processing in vitro. Dicer homologs exist in many organisms, including C. elegans, Drosophila, yeast, and humans. Dicer is a conserved protein. In Dicer's domain, it's a ribonuclease RNA family with four distinct domains: amino terminal helicase domain, dual RNA motifs in the carboxyl terminal segment, dsRNA binding domain, Has domain, etc. Now, RISC is RNA-induced silencing complex, which is a large 500 kilodalton RNA multiprotein complex which triggers mRNA degradation in response to siRNA. Unwinding of double-stranded siRNA by ATP-independent helicase. The active components of a RISC are endonucleases called Argonaut proteins which cleave the target mRNA strand. Now, what is the steps of RNA interference pathway? dsRNA is sliced by an ATP-dependent ribonuclease, sliced into short interfering RNA, siRNAs. Duplexes of 21-23 nucleotides bearing two nucleotide 3' overhanging ends. siRNAs are transferred to a second enzyme complex designated RISC. RNA-induced silencing complex, siRNA guides RISC to the target mRNA, leading to its disruption. The antisense strand of the siRNA is perfectly complementary. If you talk about the RNA interference pathway, AsRNA are chopped into short interfering RNAs by dicer. The siRNA dicer complex recruits additional components to form an RNA-induced silencing complex. The siRNA unwinds, and the unwound siRNA base pairs with complementary mRNA, thus guiding the RNA prime machinery to the target mRNA. Now, the target mRNA is effectively cleaved and subsequently degraded, resulting in gene silencing. So, these are RNA intervals between one of the hottest fields of research in the past decade, and so these are some of the things like pre-miRNAs, because miRNAs are sent to the cytoplasm from the nucleus. They generally double-stranded and loop back on themselves in a hairpin structure, even if not every letter matches perfectly. Okay, so precursor mRNA is said to cytoplasm of the nucleus. They are generally double-stranded and loop back on themselves in a hairpin structure, even if not every later matches perfectly. shRNA short hairpin RNAs. Okay, so they are even if not so shRNA is short hairpin RNAs and can be coded by the DNA introduced into a cell. They also loop back on themselves in a hairpin structures and represent one way of using RNA interference for research and therapeutic purposes. These RNAs double-stranded sequences of RNA of various lengths that can be given to the cell to initiate RNA interference. This is another way of initiating the RNA interference response in animal cells for research or therapeutic purposes. The RNA molecules that have been loaded into the RIAC find the target sequence on a messenger RNA. RIAC cleaves the messenger, destroying it when the target sequence matches perfectly. For miRNAs that don't match the target completely, the messenger is repressed and sometimes degraded by different enzymes. So all three double-stranded RNAs can be processed by dicer, a protein that cuts the strand down to roughly 21 letters long and prepares it for RNA interference by loading into a complex of complex proteins called RISC. So some RNAs that have been processed by dicer gain access to the nucleus and seem to target gene promoters. They can stop messenger RNA from being produced by the gene, but the process is unclear. Even more unclear is whether some short RNA sequences actually turn genes on, a process called RNA activation. Then pre-miRNA, primary miRNAs are transferred from the genome and processed by proteins to form pmiRNAs. Okay. So then we talk about nonsense mediated decay, which is basically cellular mechanism of mRNA surveillance that functions to detect nonsense mutations and prevent the expression of truncated or erroneous proteins. And NMD is triggered by exon junction complexes, components of the assembled RNP that are deposited during pre-mRNA processing. Antisense RNA technology blocks the activity of the mRNA in a stoichiometric manner. Antisense RNA has a opposite antisense RNA has a opposite sense to mRNA. The presence of complementary sense and antisense RNA in the same cell can lead to the formation of a stable duplex which interferes with gene expression at the level of RNA processing or possible translation. this technology widely used in plants for gene inhibition. So advantages of gene silencing: down-regulation of gene expression simplifies knockout analysis, easier than use of antisense oligonucleotides, siRNA more effective and sensitive at lower concentration, cost effective, high specificity middle region 9 to 4 are most sensitive. With siRNA, the researcher can simultaneously perform experiments in any cell type of interest and can be labeled ease of transfection by use of vector. Blocking expression of unwanted genes and undesirable substances, inducing viral resistance. Powerful tool for analyzing. Unknown genes in sequence genomes and useful approach in future gene therapy. Oligonucleotides can be manufactured quickly, some within 1 week. The sequence of mRNA is all that is needed. Now, what are the disadvantages of gene silencing? High pressure injection and electroporation can cause significant injection damage to the integrity of the normal tissues and organs and thus preclude the utilization in a clinical setup. Liposomes or cationic encapsulated siRNA may also be toxic to the host and may cause severe host immune responses. Other emerging strategies include chemical modification of siRNA molecules and encapsulated with different molecules are still in their infancy and need to be thoroughly investigated before use in therapeutic applications. Specific gene silencing using RNA interference in cell culture, cancer treatments. RNA interference has been used for applications in biotechnology, particularly in the engineering of food plants that produce lower levels of natural plant toxins. Such techniques take advantage of the stable and heritable RNA interference phenotype in plant stocks. For example, cotton seeds, modulation of HIV-1 replication by RNA interference, small RNA and its application in andrology and neurology, and developing technologies for epigenomic analysis and clinical application of molecular diagnosis. Currently, there are at least 6 oligonucleotide drugs inducing RNA interference for illness during cancer. So, inclusion is the epigenetic regulation of gene expression and widely used in agriculture and in biotechnology. Besides the all types of gene silencing, the RNA interference is the most important post-transcriptional gene silencing. Okay. Coming back to dicer, let us show you what dicer is. So it's basically double-stranded RNA triggers processed into siRNA. So enzyme RNA is C3 family, but it is the dicer family, which is positive enzyme, no larger intermediates. Dicer family proteins are ATP-dependent nucleases. These proteins contain an amino terminal helicase domain, dual RNA C3 domains in the carboxyl terminal segment, and these RNA binding motifs. They can also contain a PAS domain, which is thought to be important for protein-protein interaction. Dicer homologs exist in many organisms, including C. elegans, Drosophila, yeast, and humans. Loss of dicer, loss of silencing, processing in vitro, developmental consequence in Drosophila and C. elegans. So we have talked about all of these, all the different types of RNAs. Okay. Coming back to the question that we started off with today, mRNA maturation is dependent on dicer. Okay. Anybody has any question related to this, or you can ask now, or if not, then we can conclude today's session. Anybody? Sorry, nobody. Okay, so if nobody has any question, we can end the session for today. I'll upload these slides. I hope I was audible throughout the session.